Thank you for your participation in evolutionary developmental biology this semester. I thought it would be a good idea to close the semester by looking back at how evolutionary developmental biology functions sort of as a subdiscipline in biology. So the idea here is let's do a quick match one item from column A and column B. This is in the context of looking at fetal hemoglobin. So the fetus of a human has a hemoglobin protein that basically is more attracted to oxygen than the mother's hemoglobin so that it can get enough oxygen out of the mother's bloodstream. And these 5 bullet points then are representations of different questions these 5 different disciplines might ask. So how does fetal hemoglobin have increased oxygen affinity is something that a biochemist would ask. Something about the chemistry of the hemoglobin protein and how it changes compared to the maternal hemoglobin to have this function. How did the fetal hemoglobin gene arise during the evolution of mammalian pregnancy? That is certainly an Evo-Devo-like question. So we've got the development part related to the fetus, and we've got the evolution part related to how did this gene that caused a change evolve? How is the hemoglobin gene transmitted from generation to generation? Definitely a genetics question. How is fetal hemoglobin production expressed only in red blood cells and turned off at the appropriate time is a straight up development question. And then, well, we're left with ecology. How is it that the oxygen affinities of different populations influence the altitude at which they live? So that again should highlight for us how evolutionary developmental biology fits into biology in general as a discipline. In this semester, we've looked at several different questions, and we've looked at examples of these. So we've looked at how cells know when or where to divide or stop dividing. And although we didn't specifically discuss this question this semester, let's talk about it briefly. So given exponential growth, one cell divides and becomes 2 cells, and 2 cells divide and become 4 cells. Why doesn't the human body have exactly 1 times 10 to the 12th cells after 40 rounds of cell division? Well, it turns out humans aren't eutelic. Eutelic organisms have the exact same number of cells. They undergo development so rigorously and so identically that each individual has exactly the same number of cells. These tend to be smaller organisms than humans, like C. elegans, the worm. But so the reason that the answer is we don't all have exactly the same number of cells is we are not a eutelic organism, and also there's apoptosis, or programmed cell death, and that kills a different number of cells depending on the individual. We've also talked about the concept of differentiation, how a single genome that's present in all of our cells, or in all of the cells of a chicken, say, generates so many different cell types. And we've looked at transcription factors and gene cascades, and how different cells turn on different transcription factors that go on and turn on other genes, transcribe other genes, and so forth. We've got the concept of morphogenesis. How do cells interact with each other and produce distinct structures in the correct locations? For example, how is it that most organisms have 2 eyes and not an extra eye? And in this case, we looked at the concept of cell-cell signaling and how cells can interact and communicate with each other to ensure proper development. We've looked at the concept of regeneration, like how can some vertebrates even, like axolotls, regenerate lost limbs and others can't. And in this case, for example, we looked at planarian regeneration to understand how cell-cell signaling allows for regeneration. We've talked about reproduction. For example, how a single-celled embryo, right after the sperm-the-egg contact and fertilization occurs, how does that develop? How does the single-cell embryo produce differentiated cell types? And although we didn't talk about this latter point in gray, how the germ cells of the new organism get segregated away from the rest of the somatic tissues of the developing organism. We did sort of look at this when we talked about and read about slime molds and how communication between cells helps some cells become the germline, and how some cells sort of give up their capacity to be the germline to become somatic-like stalk cells and help the population continue to exist and to spread. We've looked at lots of, especially recently, developmental questions about evolution, like the fact that these are two mammals. One flies, one lives underwater, neither of which are sort of typical mammals. So how did this sort of variety arise? We definitely looked at the evolution of feathers versus scales. We've looked at eye loss in cavefish. We've looked at traits in stickleback fishes. And we've answered these questions. So finally, let's talk about this semester's final exam. It's worth 10 points, which is a little over a letter grade's worth of points, 12.5% of your total grade. And it's due to me via Canvas no later than 5:30 p.m. on December 16th, which is the day and time that we would be having this exam in person. It's going to be a written final exam, and this is the rubric for it, which will be also available on Canvas. I'm looking for clarity in your writing. That is, that the points you make are clear and logically supported. I'm looking at the structure of the writing, that is, the beginning with an introduction, stating a goal or hypothesis, then providing an experimental design, expected results, and a conclusion, which we'll get into a little bit more on the next slide. Most of the points are available for scientific accuracy and detail. That is, you can't just regurgitate words in this final exam, but do it clearly and in the right order with good spelling and grammar and providing citations, and pass. The details you provide need to be not only appropriate to your proposal and also explained in sufficient detail that I can tell that you actually understand that the experiments and analyses you're proposing are actually accurate and appropriate to address the goal or hypothesis you state. So with that, here are the actual instructions. Pick one paper that we read in class that is not the paper that you presented, and write an approximately 2-page proposal. Use as much space as you think you need to fully address the prompt, but I suspect you should be able to do it in about 2 pages. Give a brief introduction to the background of the topic. Start broadly, so start with the big picture evolutionary developmental biology question, then narrow to summarizing the hypothesis, results, and conclusion from that paper you've chosen, and that will comprise your introduction. Then the rest of the exam is a proposal, and that would be based on that manuscript that you read. What is the very next experiment or experiments that you think should naturally or logically happen next, with emphasis, the immediate next experiment or experiments that would continue that line of investigation? Make sure that you have a hypothesis, that you include the experimental design, and this should include our justification of the choice of organism or organisms, the detailed experimental methods, as detailed as you can get. Specifically point out the control experiments you will conduct and how they act as control experiments. And make sure also to indicate any methods you use to analyze data. For example, are the data going to be collected visually? If so, how will you visually interpret the data? Are there going to be quantitative measures? That is, are there going to be numerical values? How would you compare the numerical values? What are you going to be looking for to know whether or not your hypothesis is supported or not? Likewise, I also expect you to provide a projection or a guess at what the results would look like if your hypothesis is supported. So I do expect you to come up with some example data. It will be fake data, not real data, but either provide written descriptions of visual data that you would expect to collect, or provide a sample table, for example, of numerical data that you think you might collect. that would, again, support your hypothesis. And finally, conclude by summarizing how this research trajectory, or the series of experiments, both in the paper and the one you're proposing, relate to one of the big questions of evolutionary developmental biology that we've discussed this term. Thanks, it's been a pleasure being with you online this semester, and I hope you got a lot out of this class.